Well, I welcome you to the last and the lucky 7 session here. My name is Kris. I'm a medical oncologist at Memorial Sloan-Kettering. And I have the daunting task of filling in for Dr. Marks today. Actually, no one can really fill in for Paul Marks, those of you that know him. He is a force. But he gave me the opportunity to speak to you today, and I appreciate that. To be in this place with the research budget you all control is an extraordinary opportunity. And I think all of you almost have a budget more than the National Cancer Institute, so I appreciate having you here today. I was struck by a small article that appeared in Cancer Discovery a few weeks ago, and I'm just going to quote from the table. And this came up in one of the earlier discussions. Although spending on medicines in the United States declined by 3.5% from 2011 to 2012, and that was brought up earlier, spending on oncology drugs continued to rise, with more money spent on these drugs than any other type. So I guess I don't need to tell you that this is a huge part of your budget. The actual spending went from $24 billion in 2011 to $25.9 billion in 2012. And the greatest increase in oncology drug spending is in that targeted space. So I think I see that as a tremendous opportunity, having society investing that much capital in oncology drugs. I also will have to caution all of us here that it's a tremendous amount of responsibility. Although many have heard the saying that to those to whom more is given, more is expected. And I think more and more, and we are going to ask to be stewards of the money that we are collectively given, both from our sponsors and both from our nation and world, and being given that $26 billion a year to use properly, I think is a very daunting task. And I think we all bear some responsibility in making sure that money's used well. A couple of other issues that came up, and Kenneth Frazier, who was here a second ago, mentioned earlier how we are going to need real innovations as time goes on. That it's not just an innovation, incremental is important, but I think society is going to look to us to have advances that are really game changers, if you forgive that probably overworked term. And I'd put it to you that the number one quality measure in oncology is cure. I know that sounds crazy. You know, people talked about hitting for the fences. But I'm a medical oncologist, and I missed the morning session because I was attempting to serve as a medical oncologist. But what patients want is to be cured. And when you look at the people that regulate us, that oversee us, they are more likely to be patients than healthcare providers and healthcare innovators. And the first thing they're going to look to is cure. And they're going to say, why aren't you curing more people? I know that's a lofty goal, and when you say that it's a little unrealistic sometimes, but I do think that our patients and the people that oversee us are looking for our ability to enhance cure. Another thought is that we're focused today about drugs and oncology agents, but you heard this statistic earlier today that 70% of people that have cancer now are cured. Well, they are not cured at no cost. A huge amount of societal and cost goes into curing those patients. And when you look at the total cost of a person with cancer, drugs are a tiny percentage of that, 10-ish%, and probably people in the room here know much better what the actual number is. But 20. When people look at the cost of oncology to our society, and also for an individual patient, the cost of drugs is a small issue. And I ask you in that cost setting to say, what is the real benchmark? Well, the truth is the benchmark is cure and survival. And I'll put it to you, the most cost-effective treatment is the one that improves survival and leads to the chance of cure the most. It's benefit over time. And I think a lot of that gets lost in the kind of siloing of one area of oncology care to then as another. I think oncology drugs are an easy target. Frankly, probably radiation oncology is an even easier target. Those machines are big, they're expensive, they're easy to find, and they're heavily regulated. But we need to focus in on the bigger issues. And I think there are many opportunities here, and frankly, more opportunities than not, or I wouldn't be a medical oncologist. We're very fortunate today to have this panel because I think they are exactly where the field is right now. They have lived through many of the challenges, they've lived through many of the successes, and I think in their hands, they have the research, the technical prowess and capability to take forward, I think, some of the innovations in this field and the things that are going to shape treatment in years to come. I'm going to ask each of them to give a short few-minute description of what they think is the greatest opportunity, and then also the greatest challenges here. And I'll start with Herve Hoppenot. I've known Herve for many years. He is now the president of Novartis Oncology, and we worked in the anti-cancer field for a long time. But I think Novartis is a particular example of how they had to deal with one of the wonders of oncology. And I'm sure you heard that this morning about how when you discover a very potent addictive oncogene and you have a very potent drug, you can be a total game changer. And how even a small market can become a great opportunity for a pharmaceutical company. And Herve, we'd ask you to speak first about what you would see as the greatest opportunity right now in the area of oncologic drugs.
I would say, thank you. So I would say the greatest opportunity in my mind is what was said by the previous panel, which is that cancer is very simple. No? Because everybody was speaking about cancer as if it was already solved and that there was no problem anymore. We understand it, and we know exactly what to do. And for Alzheimer, we still need to learn from there. So I think it's probably the first time I hear that we are somewhere dealing with a very easy problem to solve. But the reality is that we are exactly there. I mean, we have seen over the years where the past 20 years, let's say, watching what is happening in the field of cancer, that there was a little bit of an ice age, literally, where things were moving very slowly, where there was a lot of very marginal moves, and suddenly it was probably 10 years ago, maybe it's a 10-year anniversary when Herceptin on one side of the world and Glivec on the other side sort of changed the way people were thinking about cancer. And 10 years later, we are literally in a situation where we feel we have just invented the internet. And we need now to develop the applications or something. I mean, we have this enormous amount of information coming from tumor genetics, understanding what they are. And it's very messy. It's an enormous amount of information that is available. But we see already on lung cancer where Dr. Kris has been at the forefront of bringing targeted therapy to lung cancer with Tarceva and Iressa, and now, 5 or 6 or 10 or 15 or 20 drugs that are available for these patients that you can include in clinical trials. And it is an amazing change. It's absolutely transformative. And it is, the world we are in today, where publications are coming every week, every month, and the publications are basically connecting the dots, which is taking patterns of gene signature and trying to connect them with targeted therapies that we have somewhere in and our includes academia, includes industry, includes many parts of the world, somewhere in the world where we have therapies that can be used for this very specific group of patients. So it's becoming something that was very messy is becoming very precise. We speak now about precision oncology, which is also like something that was almost an oxymoron in the past, because everything was relatively imprecise for pharmaceutical treatment of cancer. And we see new technologies with immunotherapy, and Dr. June will speak about it in a specific way, and maybe also in the industry where we have completely new technologies that are also changing the whole thing. So my view in terms of opportunities is that there are so many of them that we don't know exactly where to look, because there are ways now we can think about changing the life of patients with cancer in a way that was just unthinkable a few months ago and certainly a few years ago. Now, in terms of what it what it does as a challenge, one of them is the practice of medicine. And it sounds like very odd to speak about it, but there are probably in the world a number of physicians, I don't know how many there are, 200,000 who are treating cancer patients around the world in all of these countries. And none of them has been trained to these new technologies that are emerging. And when you think about it, it's not a small issue, is that there is a whole new field that needs to be put in place in the healthcare system, which includes molecular diagnostics. So how do you manage a situation where you need tumor biopsies to be transported, analyzed reliably with the good tools, and then the result to be given to physicians so that they can treat their patients the right way. And it sounds very trivial, but when you look at it, sitting on the moon and looking at the Earth there, what you see is that it is really, we are very, very, very far from there. And the patient Alpha, who is just diagnosed with cancer somewhere in the world, unfortunately, today, is not always facing a healthcare system that has been organized for this new vision about cancer. And it's not, maybe speaking from Sloan-Kettering, it sounds a little bit of a triviality, but in fact, it's a real problem around the world. The second thing, which is just a comment on what it means for patients, for people who are fighting cancer, is an interesting twist, is that access to clinical trials is probably, in many cases, not all, but in many cases today, the best option. Because the new drugs that are under study are, in many cases, the best available. And you can think of PD-1 for melanoma, or there is a lot of non-Novartis drug I can speak about, a few from Novartis also, but it's really a situation where we have put in place at Novartis a system where we try to help our associates who have a family member who has cancer. It's a very simple notion. And what we did is really ask them to contact us if they wish, and then we would put them in contact with the best possible treatment environment. And what we found is that in many cases, the best is in fact participation to clinical studies. Now, as cancer is becoming a very rare disease because we are cutting, every cancer now is very personal. Lung cancer is 200 different diseases, 150 different diseases. How to run clinical studies in a large scale so that each patient will have access to the latest technology is in fact a very significant challenge in terms of regulation, in terms of IRB processes, and how we can do that on a large scale around the world. So that, obviously the big opportunity is in front of us, how to take advantage of it and take responsibility for all the resources that are dedicated to this field is certainly not yet fully it's not yet fully organized, but I must say between academia and the industry in general, there is more and more cooperation that are really helping to make it happen.
So we'll get back to the challenges that you raised. I'd now like to introduce Dr. Carl June. He is the Director of Translational Research at the Abramson Cancer Center at the University of Pennsylvania and a leader in harnessing a untapped and yet obvious way to fight cancer. Dr. June?
Thanks, Mark. My perspective from an academic immunotherapist is that we've hit a tipping point now where, for many years, cancer immunotherapy it's not a new idea, but it just had disappointing results. We now know a lot of those reasons, and that was that we didn't understand the cancer biology as well and the intricacies of the immune system. And in my own particular field of giving engineered T cells, we had, I think, a real sort of aha moment back in 2010 when we first were able to treat people with engineered T cells that address a central problem in tolerance, in the immune system, which is tolerance, where the immune system really usually gives up on fighting against the cancer. And so now it's possible to make cells that will continue to divide in the patient and attack specifically the tumor. The initial 3 patients that we treated, those patients that went into complete remission remain that way from a single therapy. And we've now treated 55 patients, including 20 children with refractory acute leukemia, and half of those had already failed a bone marrow transplant. So they actually were, completely at the end of the line, and we have an 80% complete remission rate. So it's held up in both acute and chronic leukemia. And we see enormous opportunities now in, for instance, combining these with the checkpoint inhibitors, other ways, because in preclinical studies, they are synergistic. So now we're at an issue really where it's a whole new kind of therapy. Herve was talking about we have to educate the public and physicians about how immunotherapy can be combined with new kinds of immunotherapy. I mean, I think the goal that this field has now is to go to a place where there is no chemotherapy, and it will be targeted therapeutics, whether they're antibodies, engineered cells, and targeted small molecules. So I think the principles are there now, but the devil is in the details. In my particular field, it's how do you do something that's done at a boutique center? So our therapy evolved completely in the academic arena with no involvement in the pharmaceutical industry until just last year when we partnered with Novartis. And the issue now is that it needs to be, it's not a research issue anymore, it's now an engineering issue of how do you scale this up. And that's what the pharmaceutical industry is going to have to do, is to make this automated and not require sophisticated cell culture approaches that we currently use.
And I'd now like to give the opportunity to speak to Ira Mellman, who's the Vice President of Research and Oncology at Genentech. I think that those of us in the oncologic research field see Genentech as perhaps the treasure trove of molecules to advance the field of oncology. And again, I'll repeat it, to those to whom much is given, there are great responsibilities with it too. And I'd appreciate to hear your thoughts about what is, you think, the greatest opportunity we have right now, the keeper of the treasure.
Okay. Responsibility that is unavoidable is certainly acceptable, or accepted, has to be accepted. And certainly I feel that we and the field in general are in an incredibly good position to take advantage of what I think is the clear opportunity at the moment, which is the application of activating and manipulating the immune response to generate patient benefit. And I think this is not just an idea that comes from any one of our laboratories anymore, including Carl's, although he was a terrific leader in this. But this is really a feeling that emanates directly from clinical results, not only from cellular therapy, but as has been alluded to a couple of times already during this forum. New antibodies, such as the recently approved Yervoy, but also antibodies that attack a second area of immunosuppression of T-cell responses. These are antibodies to PD-L1, which is what we have in the clinic, as well as antibodies to PD-1, which various groups leading the way are Bristol Myers and Merck. The type of benefit that you see for cancer patients who are treated in these ways is, I would say, unprecedented. And it's not just about melanoma anymore. I think probably a very important inflection point occurred in solid tumors. And excuse me, I'm not going to speak about hematologic tumors. An important inflection point occurred just last spring at ASCO when a whole trove of results, well, still very early, but the fact that they came from so many different sources with so many different agents, basically saying the same thing again and again and again, that this is a basic biology, a basic part of cancer biology that can be manipulated to a greater or lesser extent, perhaps based on indication, perhaps based on patient. But nevertheless, we find, for example, in our trials, perhaps our best responses in lung cancer, particularly if we subdivide the patients based on the presence of a diagnostic marker, which in this case is expression, a very simple one, just expression of the target of the PD-L1 antibody. We find just absolutely astounding responses, 60, 70, 80% responses in this group. Admittedly, it's a small group, but nevertheless, it defines, I think, the biology in about as clear and a compelling fashion as you can imagine. Now, to the extent that all cancers have alterations, some genetic, some epigenetic, that yield neoantigens or antigens against which the immune system can, in principle, respond or can be convinced to respond, then, again, in principle, if we are smart enough at understanding and applying the biology, we can figure out how to press the right levers and turn the right knobs so that we can achieve this type of benefit for a very, very broad array of cancer patients. And I think almost in contradistinction to the way we have been progressing in the field up until now with ever more detailed and beautiful and elegant genomic analysis of tumors, defining ever smaller subgroups, which of course creates headaches for me now that I'm at a drug company. This changes that paradigm entirely, because you can have one drug that conceivably, if the biology is really as it seems, and as a general biology, can be applied not only to smaller and smaller patient subgroups, but actually to larger and larger patient subgroups when you can identify when these drugs can be used. So I think the challenge is how to reorder our priorities or to align with that opportunity. And we'll maybe come back to that a little bit more, but maybe just let me give you one other opportunity, which is in the area of combinations that Carl has alluded to. There are two possibilities here. Near in, you can imagine combining two different types of immune therapies that attack two different elements of the immune response. This has early steps in doing this have been done. It seems effective, but it also seems to be quite toxic. And I think, as I said, this is a manifestation of us being at the beginning of a story rather than at the end of the story. The second, though, is to leverage just the fantastic amount of information that's been gleaned over the last 2 decades, basically, in understanding cancer genomics and the generation of targeted agents which are specific to known driver oncogenes, or at least contributory oncogenes. What we need to do is understand how best to put these two things together. And unfortunately, we don't really know very much about it, because pretty much all of the work that has been done to date, say on how PI3 kinase inhibitors or MAP kinase inhibitors work, has really focused entirely on their effects on tumors and not their effects on the immune cells, whether we're trying to activate them in situ or provide them by adoptive transfer after genetic manipulation. So I think a whole other field is opening up, which is in itself going to be very, very interesting and exciting cell biology and biochemistry that's really going to deal with just how is it that cancer drugs really affect the immune system. And I can tell you that from what we know so far, the results are entirely unpredictable. You have to go through and look at them, each and every one, in a very, very distinctive fashion. So one example, you might expect that platins, which are terrible, horribly toxic drugs, would be the worst thing you could possibly combine an immunologically active agent with. Well, in actual fact, under certain circumstances, they can not only combine, but they can be synergistic dramatically. These are all preclinical studies. With clinical studies are now just getting underway. I don't understand why that's the case, but it's going to be important to understand why that's the case. And so all of this stuff comprises, I think, one of the most exciting opportunities that we have before us. And again, why I feel so positive about this, and I suspect all of us do, is because these are not based on laboratory studies. These are all being driven by what we are seeing in these very, very early days from the clinic. So the bench to bedside or bedside to bench has no meaning anymore. In fact, the bench is the bed, and we should just get used to that because it's how it is.
A lot of it. Got all that enthusiasm, right? You're going to get even more from me. I think why Dr. Marks invited me today was probably to tell this story. You know, I'm like a chemotherapy guy. Actually, I made my living giving cisplatin. And if you pull my CV, you'll find plenty of papers on that. But I was converted. I've now gone molecular. And what brought me to that was the experience in discovering the mutations in EGFR that underlie the sensitivity to agents like alatinib, gefitinib, and afatinib. And the story, to me, has 4 key components, and I think they're very helpful to this effort in oncology, but to other efforts as well, actually. There's a few resonances in the prior talk in the neuroscience section. I think the first thing about the discovery is that, and it was a dramatic one, there was no question to anybody that saw the benefit in these patients that it was important to the field and to the patient. And by the way, that included the regulators. And when the regulators see a dramatic benefit, they have a totally different view of the package. And another example is crizotinib, the drug for ALK-positive lung cancers. That was approved as the first approval on 256 patients. 256 patients. And why? No one doubted the benefit all along the line. Why? What was the cause of the magnitude of the benefit? It was understanding the biology. We knew that these, be it a mutated EGFR or a rearranged ALK, it drove that cancer cell. That cancer cell was addicted to the action of that oncogene. And by disrupting it, there was a dramatic benefit. There was a dramatic change in that cancer cell's growth. So it was biology. And I think that was an important point echoed, at least in the last session and the one before. The second thing is we had the technology to find that target, that laboratories all over the world. I used to joke that we didn't need Harold Varmus to find the mutation in EGFR. Anybody that took a course in molecular biology, I think the hardest part of the whole thing was to send away for the right primer to find that mutation. And that's really all it took. And it's also a very robust test, which is really very important. And to all the talk about biomarkers, you need a very robust test. And technology is just taken over right now because not are there only robust tests for that particular gene, but mutations in that gene, but there are very robust multiplex panels that can not only look for that gene, but in tiny amounts of material give you information that's extraordinarily accurate for literally hundreds of genes. The next part of this, and this is a subtle difference, but this came up in the last talk too, is finding the right patient. Now I don't want to be disrespectful to my pharma colleagues, but it's still today that if you're developing a new PI3K inhibitor, you are not necessarily giving that drug to a patient that has some demonstration of an abnormality in that PI3 pathway, be it upregulation, be it mutation. That is not yet the standard of care, and I could pull some papers recently about that being the case. And you heard the story in neuroscience too, that we gave an agent that was targeting a certain protein abnormality in Alzheimer's to people that didn't have that protein abnormality. So, one, I guess, corollary of the oncogene addiction story is if you don't have the target, your cancer cell is not addicted, that drug is not going to work. It's going to have a very tiny benefit, which frankly society is not going to allow us to pursue anymore. People don't want that, and I don't think they're going to give us that $26 billion to do that as time goes on. And the last thing that we had was you. I mean, I'm talking about the folks that create these drugs, that know these targets, that create extremely specific and extremely potent drugs to block, and effective drugs to block these targets. All four of those things happened in that EGFR story. They've happened again in the ALK story. They've happened again in the BRAF V600E story in lung cancer, and also played out in melanoma. And that has truly, truly influenced me. And I see that as our greatest opportunity. Does the oncogene story answer every problem? The answer is no. But it's a tremendous platform. And for those patients where we do find that, like in chronic myelogenous leukemia, and probably the best example still is that, the use of imatinib there. But that is a great platform to move forward. The second thing, and by the way, one of the biggest criticisms of it is that these patients eventually develop acquired resistance. But what happens then is that the process just restarts. When that happened, scientists quickly said, well, we know that this drug does this. The patient now is refractory to the drug, is probably the wrong term. What are the causes? They went looking for it. They found a second site mutation. And then within months, actually, or years, there are four drugs now approved for chronic myelogenous leukemia, in addition to imatinib. And it's because once you know the biology, because of you folks here that have the unbelievable capabilities and libraries of molecules, that you can translate them into a new development. I guess the most amazing thing, and I think there was a little echo of it, is the pinch me thing. I've struggled and worked hard in the field of lung cancer over many years, and no one has ever said we're a model for anything. And in many ways, we shouldn't be a good model, but people are very, I used to think leukemia was the model, by the way, as those many of us did. But now people are looking to lung cancer as that model, because I think the most important thing, it's impacted patients' lives, and it does so in a very logical way. So I think you get from all of us tremendous opportunity. But I'm going to change things up a little bit and ask people a question, though. I think we've come from the earlier sessions today. It's clear that we can't go on exactly as we're doing. We need to find ways to be more efficient, to, I think the word came up already, prioritize the various opportunities we have. Herve, Ira, you guys, you got the biggest issue here. I mean, you have so many potentially important and useful molecules in your establishments. How are you going to choose?
Want me to start? You give him time.
It's a Swiss thing, you know. We are very organized. So you start. Huh?
So while you organize, okay. Good. Since I'm from New York, I can do it. I think the biggest challenge is exactly that, because there's now a wealth of opportunity, because it was bad enough when you just had all these small molecule inhibitors and antibodies to known targets, and wondering how you're going to put those together. But now if you factor in immune modulators, that adds the number of degrees of freedom for the possible combinations to, I think, far outstrip what you could possibly do, either financially or even logistically, just on the basis of clinical studies alone. And, you know, so we have thought this through very carefully, because we don't have, even Roche doesn't have unlimited resources, I'm told. And so as a result, I think what we really come down to is to start an effort, and this is something that's really going to have to be a large collaborative effort, I think, going across not only industry, but even perhaps more importantly into academia, and especially with physician scientists, is establishing a parameter or a set of parameters to understand what relevant immune biomarkers are. How initiating the type of project perhaps similar to the Cancer Genome Atlas, except do it for immunological markers of immunological importance. By doing this, I mean, we have enough little hints from preclinical experiments that you can not only identify patient populations, but we're even seeing this already from some of our PD-L1 studies. You can identify the patients who respond, those who don't respond, and if you look at enough things, you also come up with hypotheses for why is it the ones who don't respond. And that then leads you to saying, okay, well, that means we want to try and develop something here, or we will combine with this drug when we see this particular signature. So it's all very logical, but it's all, at this point, still unbelievably crude, based on immunohistochemistry analysis of formalin-fixed paraffin sections, an occasional fluid ion microarray that you can do. We really, I think, need to have a much more serious effort in this. And I mean, to be provocative at this point in time, I would almost say that from the point of view of prioritization and patient selection, this is probably more important than continued genomic sequencing in cancer. We don't even have the techniques. At least our colleagues who are doing DNA sequencing at least can do that very well. They can write them down, and as someone said, they maybe can't interpret it. We can't interpret them yet entirely, but that will come. We don't even have the wherewithal to measure the things that we know we need to measure, and that's an enormous challenge, which of course is also an enormous opportunity, but it's going to require, I think, a rather significant collaborative effort across both, you know, academic and non-academic science.
Actually, Carl, I'd like to hear your comments on this before we get to Herve about it, particularly immune, I'll call them immune biomarkers. It has been a trail of tears. I've been quoted as saying that the ELISpot test is slightly above entrail reading. And when the technician in your laboratory goes on vacation, the test results fall off in reproducibility tremendously.
Yeah, I think this is an area of great change. We can look at correlates and lessons from vaccine field where there's been in chronic infections and trying to identify what's a positive correlation and effect. And we now know that we went down the wrong road, and a lot of times we were looking at effector cells that were end stage. And what correlates with long-term responses now is inducing T cells to proliferate and having replicative capacity and multifunctionality. And so that's a big change from the last several years. That also brings up an issue, which is that, the issue of immunosenescence, which is not classically thought of as a problem for cancer therapy when you give a drug that cross-links DNA like platinum. But if you need an immune system to work, a lot of people aren't aware, especially oncologists, that if you vaccinate someone with a standard flu vaccine and they're 65 or older, half of them don't respond. So the immune system isn't working. Maybe that's why we get cancer more as we get older, at least in part. So part of this may be regenerative efforts to make the immune system better, if we expect the immune system to control these cancers. Now, we've had 80-year-olds have complete responses to immunotherapies with very advanced cancers, so it can happen, but the younger you are, the better the immune system is. And so those are some of the correlates that I think will come out. And then we have, these issues that are going to be, companion diagnostics. There will be many bucket trials with these kind of targeted immunotherapeutics. And how
Could you explain to everybody what the bucket trial or basket trial is? I'm not sure everybody's familiar with that term.
So it won't be like just lung cancer. For instance, we're going to have next year with Novartis a trial for glioblastoma targeting EGFR V3. It's on a subset of gliomas, but it's also a subset of squamous cell lung cancers. And so an eligibility criteria would be that they have this particular mutation and not a particular histology. And so those would be very common issues. And so that's a challenge in opening the trials. Cooperative groups don't really have a mechanism many times for that. And then the FDA, would they give an indication for brain cancer, or would they give it for EGFR mutated tumors? And those are things outside of my expertise, but those questions will be raised.
But I think you embrace that idea of doing the so-called bucket or basket trial. I went to a discussion last Friday about an individual, a patient, and the issue was, did they have breast or lung cancer? And it turned out they had an amplification in HER2. So I said it didn't matter because they were both eligible for the same clinical trial, which was a basket trial for a HER2-targeted agent. But I think, Carl, you highlighted the issues there. It's tough to get everybody to that same place. Like I said, I've gone molecular here, but I'm not sure everybody has. Herve, how do you decide within Novartis? You have two potent drugs. One inhibits PI3K, one inhibits PI3K at TORC1 and TORC2 in the mTOR pathway. Which one do you take forward?
It's a good example because we are doing both, in fact.
Yes. Oh, really?
No, I think there is a myth. There is a myth I want to kill because I get this question all the time about choosing, prioritizing indication and the size. The question of the size of saying, when is something too small to be of interest to you? And frankly, people speak about Glivec and the CML story, which I think by itself is demonstrating that what was considering an orphan disease can, in fact, create a very reasonable economic model. But what people forget is that there was, at the same time and in parallel, something called GIST. And GIST is a form of gastrointestinal stromal tumors, which is a KIT mutation in some form of sarcoma. And that was so rare that, in fact, when we were trying to find patients to put in the study, we had a lot of difficulties. There were a few centers around the world. Most people were literally telling us, I heard about it in medical school, but that's, the extent of my knowledge of that disease.
It had a different name then. It used to be called leiomyosarcoma of the stomach.
Yeah, exactly, leiomyosarcoma. And what happened in GIST is that Glivec was developed. It ended up being extremely effective. In fact, it was used in the adjuvant setting, so it has cured a very large number of a small group of patients, and it has become what is today a very reasonable business, if you want to think of it that way, for Novartis. It's a $1 billion sales worldwide, so it's a few patients, but there are a big world out there. And that model is really driving a lot of our thinking, that when teams are coming to me saying, this is really a small group of patients because RAF-mutated lung cancer is only 2 or 3% of lung cancer, some small number, my answer is, remember GIST, and we go. Because if we see the mechanistic rationale for doing the study, if we see that there are patients that can be included in the study, so the feasibility of the study sometimes is the limiting factor. So the way of evaluating, screening 1,000 patients to find 5 of them can be not feasible, in fact. So the 0.5% frequency of mutation in a group of patients is creating an issue. We had the case with KIT mutation in melanoma. But in many of the other cases, we just go, and then we see. I mean, it's a little bit like a very rough way to do strategies, but it's really do the study, see what's happening in terms of clinical benefit, and if the clinical benefit is large enough, we are back to the same question. It's not a marginal something, then we go.
I think that GIST story is really an amazing one, too. I think people were very astounded to see that with a good clinical trial, that not only could this drug lead to long remissions and relapse-free survival, it appears to lead to cure. And actually with CML, too. I mean, I don't think people would have predicted that folks with CML would likely be cured by that alone. The other issue I just want to bring up again is I think, Herve's point about drug over time is very critical into the economics of it. GIST may be a small tumor. And by the way, 1% of lung cancer is 1,500 new cases a year in the US, so it's quite a few patients. But with GIST, these people take the drug every day for, now the standard in the adjuvant setting is 3 years.
3 years.
And with the chronic myelogenous leukemia, it's probably even more than that. So this kind of focus on cure, even for a tiny population, can change the economic tipping point because of the potency of the drug. And it all comes back to, again, its biology. It's a damn good drug. It's being able to find the patients and putting all those things together. And that's exactly what happened with the GIST story and the CML story. And I hope it's going to happen with the PD-L1 and PD-1 story. Ira, let me ask you a tough question. You have focused in on saying that we do have a phenotype, I'll say, that seems to predict benefit. And Liping Chen, I think, having heard him speak a few times, said if you don't have TIL and if you don't have PD-L1 present in that tumor, you're not going to have these drugs work. I think you made that point, but not every group developing this space of agents feels that way. How do you make that kind of decision to go forward and focus in on a targeted population, admittedly early on, and I think we've all agreed the assays aren't perfect at this point, but some people have made a decision to focus in based on what we have available today in the biomarker. Other people have said, we're not sure about this biomarker, let's treat everybody. How does one make that decision?
Well, even if you're establishing the biomarker, you treat everybody because you need people who are both positive and negative, and you see how it comes out in the end. I think to a large extent these things are made, the science would be clear. The science would tell you exactly what Liping says, which is that if there's no evidence for that form of T-cell infiltrate and subsequent activation, you wouldn't, why would you do this any more than you would give Herceptin to a breast cancer patient who did not have amplified HER2? If the target's not there, why would you do it? In this case, it's reflective of a biology. So we make our decisions primarily based on what the science tells us to do. So from the very beginning, we thought this is likely to have a positive correlative value that may help us identify the patients who are most likely to benefit, may cut our development costs, may make an ultimate drug, should it be approved, more acceptable or more attractive to payers, because if it's one drug that has a diagnostic associated with it that gives you a significantly higher response rate than one that doesn't, and the prices of the two drugs are the same, then I would say that one is a more cost-efficient solution to the problem than the other. How it comes out, I don't know. I mean, it's a difficult diagnostic because a lot of times expression of this particular marker we're discussing, PD-L1, is focal within tumors. And so if all you do is stick in a needle aspirate for your diagnostic test, you stand a reasonable chance of missing it. So that means you have a risk of a high false negative rate. And that is significant, I think, in everybody's hands, regardless of how good the diagnostic test is. You may still, people you call negative, say in something like melanoma, still respond at a rate of 10%, which from a historical perspective in melanoma, that's still pretty good.
Well, except as I think somebody mentioned this morning, the game has been changed there too, in melanoma.
Oh, no, it has. But if you look back in terms of why people were making the decisions, I think, basically what it boils down to is these are strategic development decisions that individual companies have made based on where they were in their clinical program with respect to drug development as compared to where they were with their diagnostic. If they were behind with the diagnostic, they say they don't need it. If you did it at the same time, you say you do need it, and the experiment will be done, and we'll see what the result is.
But it is possible to go back too, right? I mean, you could have a focused initial development plan, and then you may look for people that have less robust expression of that target or no expression of that target after you have that initial approval. Is that workable, or is that crazy?
No, that certainly can be done. Yeah. No question about it.
Herve, do you want to make a comment on that? You have to make these same kinds of decisions.
About the subpopulation in general?
Yeah.
Oh, no, there is nothing, as I said, I mean, it is clear that if you look at the sequence of events in a drug development, it's always better to start with a small group of patients where the effect is the highest, because it will accelerate your development. It will give you first the signal that you have a drug. Because our problem are not exactly the drugs that make it at some point. Our big problem are the drugs that are going on and on and on, and then at some point fail. Because, that's what, all of these successful drugs are paying for, all the ones that are not really successful in the process. So the sooner you can get a signal that you have a drug, and it's a fairly subtle thing. Sometimes it's a gut feeling. I mean, you can get a few patients, 3 patients, or a few very small number of patients treated with a new mechanism, and I will get phone calls of people in the field telling me we have a drug. We have a drug, and they can base that on their, just knowing. Same way when you looked at the first ERESA numbers, I mean, it felt like there was something more than the usual thing. So, if you can limit the scope of your first attempt to a small group of patients where you maximize the chance to see something, it's far better economically, and it's far better strategically, and it's far better for patients, by the way, because you are not exposing a number of people to something that doesn't work, which is also a key driver of that decision. So we would do that systematically. The first question we ask to the development team is, what is the smallest group of patients where you think the drug will do something? And it's also a negative test, because if it doesn't work in that group of patients, then you are really asking yourself, do you have anything to deal with?
But I totally agree with both of your comments, but that is not yet taken over as the norm. I think we're ready to open up the panel to questions about any of the issues that we've addressed. Yes.
Hi, the panel focused, I think, correctly on targeted therapies and personalized therapies, but Dr. Mellman did mention at one point almost the fortuitous finding in combination with a cytotoxic. So my question, as we look forward, what do you see the role for cytotoxic agents in the armamentarium?
Well, I've been asked to give a lecture at the ASCO meeting this year called Chemotherapy Here to Stay. So I invite you to come to that lecture. But your point is really an important one. When I go to a meeting, I don't know, Herve, I may have been to a Novartis meeting at one point, and they have their list of candidate drugs. They say, Dr. Kris, which is the most interesting drug? I always pick the chemotherapy, because every patient gets chemotherapy at some point in their illness. And again, when you look at the whole illness, chemotherapy is absolutely critical. And I guess the truth is, chemo, it really is a targeted therapy. It's just that we don't know the target. I mean, testis cancer is probably our most dramatic benefit in medical oncology, and even and hyped even more so on the cycle. But we don't know to this day as to why cisplatin cures these patients. It never could crack that nut. And it's a really important area, and we can't forget chemotherapy. And I think more and more we see these great synergies. I think the wonderful thing about the PD-1, PD-L1 story now is that it appears to be additive and can work in conjunction with a targeted or a less than targeted therapy. A targeted therapy, we don't know the target.
Yeah, no, I mean, that's true. And again, you can understand, and I think we need to understand these things from a preclinical mechanistic point of view. So I wouldn't make any blanket statements at all concerning chemotherapy should never be combined, because in fact, I believe quite the opposite is true. You may have to worry about dosing, sequencing, depending upon the effect of a given chemotherapeutic on lymphocytes or some other cell in the immune system. But they are powerful agents, and I think the problem is not one of saying that A and B are counterindicated in conjunction with immune therapy, but rather how is it that we can elicit the benefit or capture the benefit from both by understanding best how they work.
And just to echo your point about the sequence of things too, I think there's probably been too little investigation as to the sequencing of various therapies when they are combined. I know animal models don't necessarily lend themselves to that, but I know at our institution, Neil Rosen, David Solid did a number of experiments giving gefitinib with chemotherapy, and saw that the sequence was important and the dose was important actually. So I think that's probably an understudied area that we can reap some short-term benefit actually, because that's sort of just the low-hanging fruit, as we like to say. Carl, I'm sorry, you were going to make.
Well, I would just say one example, for instance, is and we give with the immunotherapeutics usually try to identify optimal biologic dose. So the trial's designed that way, whereas the chemotherapy, it's been giving at maximum tolerated dose. Now, when you try to combine the two, then what do you do? Well, it turns out maybe in the case of Cytoxan or cyclophosphamide, an alkylating agent, giving low doses daily really sometimes combines very well with immunotherapy. And there's very different mechanisms then on how it's working to promote immunotherapy than as a direct anti-tumor therapeutic. So there's a lot to learn there, and as what Mark just said, I mean, the animal models many times teach away from how to optimally do this, and the clinical trial is really the best way to understand these.
But again, I'm constantly humbled things that I do now that I never thought I would do 10 years ago. One is continue drugs even after patients progress. I was taught at oncology school that you would never ever do that. Why would you continue that drug? But there's been many demonstrations of clinical benefit, and particularly in the area of immune therapies. Also, that the dose in these defined populations is very different than dose in the wild type population. Just again, for the EGFR example, the mutant kinase is much more sensitive to the inhibitors we have. They're probably a log more sensitive. And pushing the dose, making people uncomfortable and unhealthy makes no sense biologically, and it makes no sense practically, because the idea is you're going to be taking this drug for the rest of your life, like imatinib in GIST or chronic myelogenous leukemia. And it just is a whole new way of thinking. And I urge you in this space to look at what's happened in oncology, because I think a lot of good lessons are learned there. And I guess, well, I'll give you a closing point in a second. Other questions? Yes.
In your opening remarks, you mentioned that patients are frequently interested in a cure.
No, always.
So I'm Lou DeGennaro from the Leukemia and Lymphoma Society. What I hear them tell me is they'd like to start talking about prevention. And I wonder if we're beginning to assemble enough information about the roots of the disease to begin to change the agenda and talk a little bit about prevention. I'm wondering if Carl or anybody else on the panel would care to comment on that?
You know, in animal models, clearly, where they're destined to get certain genetically engineered mouse models, you can show that chemo or immunoprevention can work. So you can, for instance, with overexpressed HER2, prevent them from getting mammary tumors. There are a number, and we have, high-risk clinics and so on where we know there's familial cancers, and then there's formidable challenges to doing those studies because of the duration of those trials, which may, by the time you see benefit, extend beyond the patent lives of the drugs. So that's an issue that I think needs to be looked at from a policy standpoint to encourage those kinds of immunoprevention trials. Another issue, is in immunoprevention, we look at, for instance, BCR-ABL, for instance, the Philadelphia chromosome is what is the driver mutation in CML, the drug that imatinib cures. Well, if you look at people with a sensitive assay, about 1/3 of healthy adults have the Philadelphia chromosome. So who would we treat? I mean, you would have to treat people, to prevent, but the readout's not going to be whether or not you have a Philadelphia chromosome, because many healthy people do, and they acquire that with age, but for whatever reason, it never progresses to overt leukemia. So that's another challenge as to who we would try to do to, for instance, prevent leukemia, which is something I'm very interested in, and I know many families are.
Yeah, I will also say that it's been something that I think everybody would want to do, but it's been extraordinarily hard. As far as the causes, I mean, the obvious would be cigarette smoking, and sadly, it's very difficult to make those kind of lifestyle changes and removing addictive substances from our environment. And that goes along too with industrial pollutants, which may have a role in leukemia. The other issue is in terms of prevention, and those of you that are interested in this space, two stories. If you go to the American Cancer Society dietary guidelines for supplements, there is only one level 1 recommendation, and that is you do not give vitamin A targeted compounds to people that continue to smoke. And the reason is when they did a clinical trial to give vitamin A targeted compounds as a preventative, they actually increased the rate of cancer. So it's a very sobering story. And it's very hard when you have people that don't have the serious illness. Your level of risk acceptance is really, really low. And that for an oncologist, that's the toughest thing. The other thing is when you discover a drug, and I'm not a breast cancer specialist, but I do know, I think we have 3 drugs approved now for the prevention of breast cancer in high-risk groups, and they are very seldom used, is my understanding of that. And it's very, very hard to take people that do not have the illness and to take any risk. And I think that's a very reasonable tactic. And so I guess the big thing is get people to stop smoking. That's one thing we can do, and there's no disagreement there. Yes?
I was wondering what the panel feels about the role of epigenetics in controlling or monitoring progression.
I didn't hear the question.
Oh, the question, the role of epigenetics in the oncologic process, and I'll take the next step is targeting that.
Yeah, well, it's something we have started to do because I think as you look at the more recent genomic analysis that has been done, you find repetitively, and I think this was mentioned earlier today, an accumulation of mutations that are in epigenetic modifiers. Many of these are in genes that are presumptive tumor suppressors, but not all. So IDH1 was mentioned. That's an indirect modifier because it actually modifies a methyltransferase indirectly, or demethylase rather. But there are others that affect so-called readers, which are bromodomain proteins and other things, either add or subtract specific methyl groups. So the idea is that it may be a new target landscape that's opening up. In terms of monitoring it as a function of progression, the assays are not quite there yet. I mean, short of just going and sequencing all of the genes, what may be more interesting is to sequence the histone marks and the DNA methylation phenotypes that occur in cancer as they change, and they do. But at least I'm unaware of any clear correlations yet that have emerged from that. But this is very, very new biology, and I think is very exciting.
I think today we probably haven't exhausted all the various approaches that have promise, and I think that clearly, would be one of them. I guess, Carl, you and I both, I have the same boss and your former boss. We have to mention, metabolomics, and that is a whole other area of research and potential that we have not touched on today. But I'm sure if Dr. Thompson were here, he could wax poetic on that. All right. I think we need to close the session, and I appreciate your staying and being with us here. And thank you very much. I thought the talks today were fantastic. And again, it's an honor to be with you that are really controlling so much in how our nation fights cancer. Thank you.